Okay, last but certainly not least, we have to talk about the fourth compound of life, that being nucleic acids. And I say certainly not least because life on Earth wouldn't exist without these guys. This is our genetic material. So another way to think of nucleic acids are polynucleotide polymers. And that's because the polymer of nucleic acids are called polynucleotides. Now, specifically, the most important nucleic acids we're talking about are DNA, deoxyribonucleic acid, and RNA, ribonucleic acid. And of course, we're going to spend tons of time in this course talking about these two molecules because these are the basis of life, whether you are a virus all the way up to a human or an elephant or a sequoia tree. We're all controlled by these molecules here, which are, again, just like everything else we've been talking about, incredibly simple molecules when you get down to it, composed of chomps, right? Yet incredibly complex in what they can do. So specifically, DNA and RNA are, of course, our hereditary material. Here's a space-filling model of a DNA molecule. We're going to give a brief overview of what this molecule is composed of, but we'll revisit that later on as well. So what do these molecules, DNA and RNA, do? Well, together, they control the structure and function of our cells. They're the boss. They're located in, well, DNA specifically, located in the nucleus of each of our cells. And remember we talked with proteins, I mentioned the term protein synthesis. We'll revisit that at the end of this lecture. But basically, the nucleic acids, DNA and RNA, go hand in hand with the production of proteins, and therefore the production of traits of everything that goes on in and out of our body. So how does this happen? We're going to give an overview. Later on in the course, we're going to get into the nitty-gritties of that secret of life, the central dogma, protein synthesis. So all of that, protein synthesis, is controlled by our DNA. And so the way that happens is through what's called gene expression. Now you may be asking, what in the world is a gene? I mean, you hear the term all the time. You know, you can go on ancestry.com or whatever and find out some more about your genes. You may look at your parents or your children and try to figure out what genes you have in common. But really what we're talking about is a physical thing. So in actuality, the human genome consists of about 30,000 or so, still a little bit for debate, but about 30,000 genes that actually code for proteins, which code for traits. And it's really amazing. Inside the nucleus of one of our cells, of a human cell, you can fit about 7.69 feet of DNA, if you were to line it all up end to end, squashed into that nucleus. That's really incredible if you think about it. And it's just one little segment on that DNA that makes up a gene, and it codes for a particular protein, which codes for a trait. So we can say that the unit of hereditary information is the gene, a segment of DNA that codes for a protein. Now, obviously, DNA being a polynucleotide, being a polymer, must be built of monomer components. And so what are the monomers of nucleic acids? We call these nucleotides. So that's why the polymer is a polynucleotide. The monomer is a nucleotide. And we're going to visit exactly what that looks like. So here is a nucleotide monomer. And it's, you know, whether you're dealing with DNA or you're dealing with RNA, it's the same basic components to this monomer. So here it's just giving you kind of the general geographic shapes of these components, and here a little more detail. Still not a lot of detail, but a little more detail. But in general, remember our favorite functional group, the phosphate molecule, PO43-. And I told you, don't forget that functional group because it's used in nucleic acids as well as a ton of other things. So it's so important, and in fact, being such a negatively charged compound, that phosphate is going to help give DNA and RNA an overall negative charge. And if it wasn't for that, we couldn't do all this cool CSI stuff that we can do now with biotechnology in breaking down the DNA and comparing suspects to the crime scene and things like that. So we have a phosphate group attached to a pentose sugar. That means a 5-carbon sugar. Remember, there's a carbon at each point here. And that is attached to what we call a nitrogenous base. And in one sec, we're going to go over what the nitrogenous bases of DNA and RNA are. But there's not too many of them. So if you take the pentose sugar, the 5-carbon sugar, which you can see it spelled out right here, and the nitrogenous base, those two things together make up the nucleoside component of the nucleotide. Nucleoside component of the nucleotide. So the whole thing is called a nucleotide. This part's called the nucleoside. And here it spells it out for you. There's your phosphate group attached to your 5-carbon sugar attached to your nitrogenous base. Okay, so that is the basic monomer of these polynucleotides, these nucleic acids. So I mentioned DNA and RNA. They sound very similar because they are very similar. And in fact, it's widely believed that RNA was actually the evolutionary precursor to DNA. But we need both of them for we eukaryotes. So let's compare RNA and DNA together and find out what the similarities and differences are. And it's incredible, actually, how similar they are. There's very few differences, and it could have been those slight chemical differences, kind of been a theme we've been talking about here, that led to the ability for the evolution of complexity of life on Earth. So let's take DNA first. So here is DNA, and most of you know DNA is a double helix, and got a fun little cartoon here. So the night before Watson and Crick discovered the structure of DNA, they went to the bar, and Watson said to the bartender, "Make it a double helix." Ha! Anyway, okay, so it's a double helix, and let's break this double helix down. What is it composed of? Well, the rungs of the ladder, so if you thought of this as a spiral staircase, the rungs of the ladder are composed of those nitrogenous bases. And for DNA, there are 4 of them. We have cytosine, which is this compound right here, and we can abbreviate that with a C. Guanine, which we can abbreviate with a G. Adenine, which would be an A. And thymine, which we'd abbreviate with a T. And you can see the similarities between cytosine and thymine and the similarities between guanine and adenine. We'll get into that a little bit more later. And there's a base pairing rule, right? So C, always, if things go right and there's no mutation, pairs with G. So C and G together, and A pairs with T. So these make up the rungs of the ladder here. If you're color coding this, notice they put the green paired with the blue to represent guanine paired with cytosine. Now, together, these are called the base pair, or the nitrogenous base pairs. Now, the sides of the ladder are made up of alternating phosphates and those pentose sugars. In the case of DNA, it's called deoxyribonucleic acid because it uses the sugar deoxyribose. So remember, anything ending with OSE is a sugar. So we have phosphate connected to deoxyribose, connected to our base pairs. So notice that DNA, being a spiral staircase, is double-stranded. So that means it turns out there's hydrogen bonds between each of these base pairs, and we're able to therefore break those weak hydrogen bonds to separate the 2 sides of the spiral staircase, and that will become important later on, as you will learn, because that's how we're going to replicate our DNA. Can't replicate DNA, you can't have life, right? You can't replicate life. Now let's look at RNA, which stands for ribonucleic acid. Notice it's not a double helix. It's single-stranded, unlike DNA, which is double-stranded. So it's single-stranded, which means it only has 1 row of nitrogenous bases here. And what's cool is that 3 of the 4 nitrogenous bases of RNA are identical to the 3 of the nitrogenous bases of DNA. So RNA also uses cytosine, it also uses guanine, and it also uses adenine, but there's a difference. Instead of thymine, it uses uracil, which we abbreviate with a U. Still a very similar compound to thymine, and I'll show you this in more detail in a little bit. But very, very similar. And the other difference is between the pentose sugars on the backbone. So it still has alternating phosphates and sugars going all the way down the backbone, But instead of deoxyribose, it just uses ribose. So a slight chemical difference in the sugars. That's it. Other than that, quite identical. Still has the same base pairing rule, C with G. And now, because it doesn't use a T, A is going to pair with U. Okay, so let's look more at DNA specifically right now. Let's look at their 4 bases. Again, A, T, C, G, adenine, thymine, cytosine, guanine. And we can take those bases and divide them into 2 categories based on their size, actually. So here are what we call the purines. And the purines are the adenine and the guanine. So if you notice, both of these are the larger kinds of bases. So they both have this double ring structure here. And look how similar. What's the difference between adenine and guanine? Well, not much. This one has a ketone group here, right? So there's a carbon double bonded to an oxygen. And here the carbon is bound to an amino group, NH2. And here there's a hydrogen versus here there's an amino group there. That's it. That's the difference between A and G. And imagine in chemical evolution, slight tweak of the functional groups, boom, completely different nitrogenous base, and we're building the molecule of life there. So those are the purines. How I remember it, my mnemonic is I say pure agriculture, right? So ag, adenine, guanine, purines, pure. And the other group are consisting of the pyrimidines. So that, of course, is cytosine and thymine. And then going over to the RNA side, uracil would also be considered a pyrimidine. And all 3 of these compounds, check them out. Single ring structure, very, very similar in composition. In fact, look at thymine and uracil, and I'll show you a picture of this in a sec. Very, very similar. They both have the ketone group right there. I mean, so similar. This one, the thymine has a methyl group there where that one doesn't. So almost identical, except for that one functional group. So again, imagine over evolutionary time, slight tweak, boom, difference between RNA and DNA, and the difference between going from a very, very simple structure, perhaps an RNA virus, to the rest of life on Earth. And checking out cytosine, very similar still, except it has an amino group instead of a ketone there, and just slight bonding difference there. Pretty cool. And this one has H's there instead of a methyl group. This diagram here is kind of cool because it shows you what the DNA molecule looks like unwound and showing you the chemical composition. So notice again, here's your pentose sugars alternating with your phosphates. So phosphate sugar attached to a nitrogenous base, base paired based on the base pairing rule, and on the other side attached to the sugar and the phosphates, and they alternate phosphate sugar, phosphate sugar. We say that the strands of DNA are anti-parallel because one goes 5' to 3', and the other one goes 3' to 5', and that has to do with which carbon is up. And then again, don't forget these hydrogen bonds that connect the 2 sides of the ladder together between these complementary bases. Pretty cool. And then over here, this is just showing you more detail of the very slight differences between the important molecules in DNA and RNA. So here is deoxyribose, the sugar in DNA. Here is ribose, the sugar in RNA. Only difference between them, boom. Deoxyribose has a hydrogen where ribose has a hydroxyl. That's it. And then here is between thymine and uracil, and again, only difference, thymine has a methyl group which replaced this hydrogen right there. So unity of life, so cool. Now a little more about RNA. So whereas DNA, there's really only one kind of DNA that we talk about much. However, RNA has 3 very distinct types, and each do a different thing, and they look very different from each other. So we're going to spend a lot of time talking about, well, all of these. One kind of RNA is what's called mRNA, or messenger RNA, and that's because it's going to take the message from the DNA out to a structure called a ribosome, which we'll be learning about soon, that will actually be the surface on which we create our proteins. So mRNA is the messenger in protein synthesis. And it's just kind of a long, snaky, single-stranded molecule that is going to carry a code based on those nitrogenous bases that are going to be written based on the order of nitrogenous bases on the DNA. So it's kind of single-stranded and snaky. So that's messenger RNA. Also involved in protein synthesis is another kind of RNA called tRNA, or transfer RNA. And I love it because it's all funky looking. It's like, to me, it kind of looks like a T. And it's because it actually, when you have the RNA strand itself, it will kind of fold up on top of each other, or on top of itself, kind of like proteins do. Remember, this is a nucleic acid, because part of it can bond with another part of it. And so it ends up forming this interesting structure. And when you learn about protein synthesis in detail, you'll learn it's the tRNAs that carry amino acids to their site to be joined to the polypeptide chain. And then the third type is called rRNA, or ribosomal RNA. It's slightly different structure. And this is a major component of those little organelles called ribosomes that I mentioned before. So 3 types of RNA: messenger RNA, mRNA, transfer RNA, tRNA, and ribosomal RNA, rRNA. Okay, so all of these, DNA, RNA, all those kinds, and proteins all come together in a beautiful display called protein synthesis. And I'm going to hammer protein synthesis into you because this is the secret of life. It's called the central dogma of biology, protein synthesis. Don't forget it. Don't forget it's the central dogma. And again, the details of it are beautiful and a bit complicated. So we'll spend a whole lecture just on protein synthesis. And but it's the basis of understanding life. It is how you bridge the gap from your hereditary material, your DNA, to the traits that actually make life life. And it's the basis of evolution. So if you want to understand evolution and how do we get the diversity of beautiful organisms that exist on this planet, you really have to understand protein synthesis and gene expression. And so the central dogma is DNA codes for messenger RNA, which codes for proteins, which code for the traits, which code for you and plants and all the other animals and bacteria and viruses and et cetera, et cetera, et cetera. Pretty much everything. So this is the basis of life. So we're going to get our DNA located in the nucleus of our cells. It's going to be transcribed with messenger RNA into a message that can be read. And from there, it's going to go out to the space outside the nucleus called the cytoplasm, you'll be learning about, into one of those little ribosomes we mentioned, which contains ribosomal RNA along with other things. And from there, it's going to be translated into a growing polypeptide chain consisting of amino acids. And those chaperone proteins are going to fold that polypeptide chain up into the functional protein. And it's that protein that can take on all those, depending on what kind of protein we just made, we'll do all those different functions that we talked about. So your traits, and putting all this together, that is how life functions. So stay tuned for the mystery of life to be unraveled. And it's an ongoing thing. This ain't your grandma's biology class because we're still just scratching the surface of the mystery of life, but we're learning more every day. So I hope learning about these 4 compounds of life has given you an appreciation for the simplicity yet beauty that leads to the amazing complexity of the millions of creatures on this planet. See you next time.